The Paracas elongated skulls. In 2004, Peruvian archaeologist Julio Tello uncovered a necropolis in the Paracas Peninsula containing over 300 elongated skulls that defied conventional anthropology. These weren't the result of cranial binding, a practice known across ancient cultures. The bone structure itself was fundamentally different. The skulls possessed 25% more cranial volume than modern humans, displaced eye sockets, and a cranial suture pattern never seen in Homo sapiens. When independent geneticist Brian Forster finally obtained permission to conduct DNA analysis in 2013, the results were so anomalous that 3 separate laboratories refused to process them further. The mitochondrial DNA sequences showed markers that didn't match any known human haplogroup. Segments of the genome aligned partially with Denisovan ancestry, but other portions contained unknown genetic signatures that database algorithms couldn't classify. The nuclear DNA revealed even stranger patterns. Protein coding genes that appeared functional but had no analogs in modern or archaic human populations. One sequence suggested a melanin production pathway radically different from any primate lineage, potentially explaining historical accounts of red-haired mummies found in Paracas tombs. Most disturbing was the chromosomal analysis. Standard human DNA contains 46 chromosomes. Preliminary karyotype studies on the Paracas samples suggested structural anomalies consistent with chromosomal fusion or duplication events unknown in human evolution. The laboratory conducting this analysis, a respected facility in Connecticut, abruptly terminated the project after the second round of sequencing. Their official statement cited contamination concerns, despite the samples having been handled under sterile protocols used for ancient DNA worldwide. The Peruvian government responded by reclassifying the Paracas skulls as national treasures, effectively ending all independent genetic research. Foreign scientists seeking permits to study the remains have been systematically denied since 2015. The National Museum of Archaeology in Lima displays several skulls, but only as examples of cranial modification, with no mention of the genetic testing that was conducted. Educational materials distributed to tourists explicitly attribute the elongation to binding practices, contradicting the morphological evidence. What happened to the DNA data? According to sources within the original research team, the full genomic sequences were uploaded to a private server before the project was shut down. That server was reportedly accessed by an unknown party in 2016, and all files were deleted remotely. Backup samples stored at the Peruvian lab were destroyed in a fire that investigators ruled accidental, despite the facility's modern fire suppression systems. The lead geneticist who coordinated the analysis left the field entirely and now refuses interviews. The Paracas skulls present an uncomfortable question. If these individuals were simply humans with modified skulls, why does their DNA deviate so significantly from known human lineages? And if they represent something else, a hybrid population, an isolated evolutionary branch, or evidence of genetic manipulation, why has every attempt to investigate been systematically obstructed? The bones remain, silent and elongated, in climate-controlled vaults, but the molecules that could explain them have been locked away in a deeper vault: institutional denial. The Denisovan hybrids of Tibet. In 1980, a Buddhist monk exploring caves in the Tibetan plateau discovered a jawbone embedded in sediment at over 10,000 feet elevation. The bone sat in a monastery for decades until 2010, when researchers from Lanzhou University obtained permission to examine it. Carbon dating placed the mandible at over 160,000 years old, but it was the DNA extraction in 2019 that triggered an immediate lockdown of all related research. The genome recovered from the Shaha jawbone matched Denisovans, the mysterious human relatives known only from fragments found in Siberia's Denisova cave. But this specimen contains something unprecedented: clear evidence of interbreeding with another hominin population that left no other fossil record. Roughly 15% of the genome came from an unidentified source that diverged from the human family tree over 1 million years ago, predating even Homo erectus migrations out of Africa. Geneticists called it a ghost population, something that contributed DNA but left no bones, no tools, no trace except genetic echoes. The Tibetan sequences revealed adaptations for high-altitude survival far more sophisticated than those in modern Tibetan populations. The EPAS1 gene, which regulates oxygen metabolism, showed variants that would have allowed sustained activity at oxygen levels lethal to modern humans, but other sequences pointed to even stranger adaptations. One cluster of genes appeared related to cold tolerance mechanisms found in Arctic animals, not primates. Another segment suggested enhanced night vision through rhodopsin modifications never documented in any human population, ancient or modern. When the research team attempted to publish their findings in Nature, The paper underwent 14 months of review, unprecedented for ancient DNA studies, before being conditionally accepted with mandatory redactions. Specific genetic sequences had to be removed from supplementary data. Phylogenetic trees showing the relationship between the Shahe individual and other hominins were required to be simplified. The final published version omitted all mention of the ghost population's potential geographic origin or the physiological implications of the novel gene variants. Chinese authorities subsequently declared the cave system a protected cultural site and terminated all excavation permits. The monastery that originally housed the jawbone was instructed to cease all cooperation with foreign researchers. A planned expedition to search for additional Denisovan sites in the region was cancelled without explanation in 2020. Satellite imagery shows construction activity near the cave entrance, possibly a military installation, though no official explanation has been provided. The implications are staggering. If Denisovans interbred with an unknown hominin in Tibet, then human evolutionary history is far more complex than the simplified narratives in textbooks. If that ghost population possessed adaptations suggesting non-African origins or radical environmental specialization, it challenges fundamental assumptions about where and how humans evolved. And if modern populations carry these genes as genetic surveys increasingly suggest, then we are all partial descendants of something science cannot yet name. The Shahe mandible remains in Chinese custody. The full genome sequence exists somewhere in restricted databases, but the story it tells of mysterious ancestors in high mountain caves, of hybridization events that shouldn't have been possible, of traits that seem engineered rather than evolved, remains officially unwritten. The bones remember what the institutions prefer to forget. The red-haired giants of Nevada. In 1911, miners excavating guano from Lovelock Cave in Nevada broke through a layer of bat dung and discovered something that would become one of America's most suppressed archaeological mysteries. Beneath the natural debris lay the mummified remains of several individuals who averaged between 7 and 8 feet tall, with distinctly red hair and skulls showing morphology inconsistent with any known Native American population. Local Paiute oral traditions spoke of the Sitaka, red-haired giants who had warred with their ancestors before being driven into the caves and destroyed. The miners had stumbled into what might have been a mass grave or a tomb. Initial documentation was thorough. University of California anthropologist Llewellyn Loud conducted the first scientific excavation between 1912 and 1924, cataloging over 60 skeletons. His field notes described individuals with excessive stature, unusual cranial capacity, and hair pigmentation foreign to indigenous populations. Several skulls showed pronounced brow ridges and elongated craniums, features rare in Native American remains. Most remarkably, tissue samples from the mummified bodies contained red hair, not faded black or brown, but genuine red pigmentation consistent with Northern European populations, yet carbon dated to between 3,000 and 10,000 years old. In 1967, a second examination was conducted by the Nevada State Museum. This time, advanced chemical analysis confirmed the hair was naturally red, not colored by minerals or degradation. DNA extraction was attempted using the primitive techniques available at the time, but the results were deemed inconclusive and never published. The skeletal remains were measured again, with several femurs indicating standing heights approaching 8 feet. Comparative cranial analysis suggested morphological features that aligned neither with Native American populations nor with any known European groups. The bones seemed to represent something distinct. Then came the disappearance. In 1979, the Smithsonian Institution requested the transfer of all Lovelock Cave remains to their physical anthropology collection in Washington, D.C. The Nevada State Museum complied, shipping over 40 skeletons and numerous artifacts. When researchers later requested access to study the collection, they were informed that the remains had been misplaced during archive reorganization. No inventory exists. No photographs of the transferred materials were kept. The Smithsonian's official catalog contains no record of ever receiving them. What DNA evidence existed before the transfer? According to a whistleblower who worked in the Nevada State Museum's anthropology department, preliminary genetic testing in the early 1970s revealed mitochondrial haplogroups inconsistent with Asian-derived Native American populations. The samples showed closer affinity to ancient European hunter-gatherers, specifically markers related to the Basque and Celtic populations. But the results were never published because, as the whistleblower claims, they contradicted federal policy regarding indigenous land rights and tribal ancestry claims. If confirmed, such findings could have complicated legal frameworks dependent on proving continuous Native American habitation. Modern attempts to investigate Lovelock Cave have been systematically blocked. The site is now on private land, and the owner refuses all excavation requests. Ground-penetrating radar surveys conducted illegally by amateur researchers in 2015 showed additional chambers beneath the known cave system, potentially containing more remains, but when they attempted to publish their findings, legal threats forced them to retract all data. The Bureau of Land Management maintains that the area contains no significant archaeological value. The Sai-Takao remain trapped between legend and evidence. The Paiute remember them, the miners found them, the scientists measured them, and then they vanished, not into myth, but into institutional silence. If the bones existed, they proved something unauthorized, and what is unauthorized in science often becomes unreal. The Starchild Skull. In 1930, in a mine tunnel south of Chihuahua, Mexico, a teenage girl discovered two skeletons buried together in unusual circumstances. One appeared completely human. The other possessed a cranium so distorted and enlarged that she initially believed it was deformed. The skulls remained in her family for decades until 1998, when paranormal researchers obtained them and began what would become one of the most controversial genetic investigations in modern anthropology. The so-called Starchild skull exhibited features never documented in medical literature: massively expanded cranial volume, shallow eye sockets, reduced facial bones, and a cranial base suggesting the brain had grown forward and laterally rather than upward. Independent researcher Lloyd Pye spearheaded the first serious scientific analysis in 1999. Standard morphological examination confirmed the skull was not the result of hydrocephalus, cranial binding, or any known pathology. The bone itself was dramatically thinner than normal human skull bone, averaging just half the thickness, yet denser with a microstructure resembling compressed fibers rather than typical osseous tissue. The eye sockets were positioned too shallow for human eyes, suggesting either a deformity incompatible with life or an alternative visual system. The foramen magnum, where the spine connects to the skull, was positioned more forward than in humans, implying a different posture or neck structure. DNA testing began in 2003 at multiple laboratories. Mitochondrial DNA extracted from the skull returned typical human sequences, specifically haplogroup C, common in Native American populations. But nuclear DNA inherited from both parents revealed anomalies that geneticists couldn't explain. Significant portions of the genome failed to align with human reference sequences. Some fragments showed closer similarity to bacterial or fungal DNA, which researchers attributed to contamination. But other sections contained sequences that matched no known organism in genetic databases. These no-match fragments were concentrated in genes related to neurological development and bone formation. The most controversial finding came in 2011, when a Canadian laboratory conducted a more thorough genomic analysis. They identified sequences on what appeared to be the X chromosome that deviated significantly from human norms. The report suggested the skull might represent a human hybrid individual, though it carefully avoided specifying what the non-human parent might have been. The laboratory's findings were never formally published. Shortly after submitting the draft paper, the lead geneticist was instructed by his university to cease all work on the specimen. The official reason given was ethical concerns regarding research on indigenous remains without proper tribal consultation. Academic pushback was immediate and harsh. Mainstream anthropologists dismissed the skull as a hydrocephalic human child, despite multiple medical experts confirming it showed none of the standard markers of that condition. Geneticists claimed the anomalous DNA was simply degradation artifacts or contamination, despite the sequences being reproducible across multiple extractions and laboratories. When Pye attempted to present findings at conferences, he was refused entry or his sessions were canceled. Funding for continued research evaporated. The skull itself became a legal battlefield, with the Mexican government claiming ownership and demanding its return, effectively ending all scientific access. What makes the Starchild skull's suppression particularly troubling is the pattern of institutional resistance combined with the physical impossibility of the official explanations. If the skull truly represented a hydrocephalic child, why does its bone structure defy all medical precedent? If the DNA anomalies are contamination, why do they cluster in developmentally significant genes? And why would authorities across multiple countries expend such effort to prevent further testing of what should be, according to skeptics, an unremarkable pathological specimen? The skull exists in storage, its secrets locked in molecules that are no longer allowed to speak, and the question it poses remains suspended in the air. What happens when the evidence refuses to fit the paradigm, but the paradigm refuses to change? The answer, it seems, is that the evidence disappears. The School Kafse hybrids. Between 1929 and 1934, excavations at School Cave on Mount Carmel in Israel uncovered 10 skeletons that rewrote everything anthropologists thought they knew about human evolution. The remains, dated to over 100,000 years ago, showed a confusing mixture of Neanderthal and anatomically modern human traits. Skulls displayed both the pronounced brow ridges and occipital bunning characteristic of Neanderthals, yet had the high foreheads and reduced facial prognathism of Homo sapiens. The skeletal structure suggested individuals who were neither one species nor the other, but something in between. When Dorothy Garrod first published her findings in the 1930s, she suggested these might represent an evolutionary transition between archaic and modern humans. But as genetic science advanced, that explanation became untenable. Modern humans and Neanderthals were separate species that evolved in parallel, not in linear succession. So what were the school individuals? In 2015, when ancient DNA technology had matured enough to extract usable genetic material from such old bones, a team from the Max Planck Institute finally attempted to answer that question. The results were never published. According to leaked correspondence between researchers, the school DNA showed a level of Neanderthal admixture far exceeding anything found in modern humans. While contemporary Eurasian populations carry roughly 2 to 3% Neanderthal genes, the school individuals showed levels approaching 25%, suggesting they were first or second generation hybrids. But the genetics revealed something even more unexpected. Evidence of gene flow from a third population, something related to neither modern humans nor Neanderthals. This unknown contributor appeared in segments controlling immune response and neurological development, hinting at a now extinct hominin group that left its mark only in these hybrid individuals. The morphological implications were equally troubling. Reconstruction of the school skulls suggested brain structures intermediate between species. The frontal lobes associated with abstract thinking and planning showed modern human proportions. But the parietal lobes linked to spatial processing and sensory integration retained Neanderthal characteristics. This hybrid cognitive architecture suggested individuals who might have thought in fundamentally different ways than either parent species, combining analytical capabilities with enhanced environmental awareness. From an evolutionary perspective, such hybrids should have possessed significant advantages, yet they vanished. In 2016, the Israeli Antiquities Authority suddenly restricted all access to the school remains. Foreign researchers who had been granted permits found them revoked without explanation. A planned comprehensive genetic survey of all school and nearby Kafse remains was cancelled. When a German anthropologist protested, citing years of approved research, he received a terse response stating that national security considerations required the suspension of all genetic work on Israeli hominin fossils. The bones were moved to a secure facility, and all previous DNA extracts were recalled from international laboratories. Why would hybrid skeletons from 100,000 years ago pose a security threat? Some researchers believe the answer lies in what the genetics revealed about modern populations. If the school Kafse hybrids successfully interbred with early Homo sapiens populations, then their unique genetic combinations might persist in modern humans, particularly in Levantine populations. Such findings could complicate the carefully managed genetic narratives around ancestry, identity, and territorial claims in one of the world's most politically volatile regions. Science and politics have always been entangled. Here they may have become inseparable. The school caves remain accessible to tourists. The story told is simple. Early modern humans lived here, then moved on. But deep in restricted archives, the bones tell a different story, of species boundaries that blurred, of hybrids who shouldn't have existed, of genes that traveled through millennia to reach us. And the silence around them suggests that some truths about human origins are considered too dangerous for open discussion, not because they're false, but because they're too true. The Tarim Basin mummies. In 1978, Chinese archaeologists working in the Tarim Basin of Xinjiang province made a discovery that immediately became classified. Dozens of mummified bodies, naturally preserved by the desert climate, with distinctly European features. The corpses had light brown to blonde hair, high-bridged noses, and skeletal proportions more consistent with Celtic or Germanic populations than with East Asians. The mummies were dressed in woven wool textiles, using patterns identical to those found in Bronze Age Austria and plaid designs matching ancient Scottish tartans. Radiocarbon dating placed them between 4,000 and 2,000 years old, far earlier than any documented Western contact with China. For decades, the Chinese government refused to acknowledge the discovery beyond vague references to ancient Xinjiang inhabitants. But in 1993, photographs leaked to international media, forcing a response. The University of Xinjiang was permitted to conduct limited studies, but genetic analysis was explicitly forbidden. When Dr. Victor Mayer of the University of Pennsylvania finally convinced authorities to allow DNA testing in the early 2000s, the results confirmed what the physical evidence suggested. The Tarim mummies were descended from a population originating in the Pontic-Caspian steppe of Eastern Europe, with genetic markers matching modern Basque and Celtic populations. The implications were politically explosive. The Xinjiang region is home to the Uyghur people, who claim the area as their ancestral homeland. The Chinese government maintains that Xinjiang has been part of China since ancient times. But if the earliest inhabitants were Europeans who established advanced settlements thousands of years before Han Chinese expansion, it complicates territorial narratives. Moreover, if these Indo-Europeans brought technologies like bronze metallurgy, wheel-based transport, and possibly even writing systems, it challenges the assumption of Chinese civilization developing independently. In 2009, genetic analysis revealed even more troubling details. The Tarim population showed signs of admixture with a now extinct East Asian group that predated known Siberian migrations. This ghost population's genetic signature suggested they had lived in isolation for tens of thousands of years before encountering the European migrants. Some samples showed tripolar ancestry European, Mystery Asian, and traces of Denisovan DNA, creating individuals with genetic combinations never documented elsewhere. The fusion implied a sustained period of interbreeding and cultural exchange in Central Asia that has no parallel in historical records. When researchers attempted to publish comprehensive genetic data, Chinese censors intervened. Papers were required to remove all references to European ancestry in the abstract and introduction, burying the findings in supplementary materials. Phylogenetic trees had to be redrawn to minimize visual emphasis on Western genetic connections. Any discussion of migration routes was forbidden. The final published versions transformed a story of European pioneers in ancient China into a vague narrative about diverse population movements. By 2015, the Tarim mummies had been moved to a new museum in Urumqi, where they remain on limited display. The exhibition labels describe them generically as ancient Xinjiang residents, with no mention of their proven European ancestry. Requests for new DNA samples are categorically denied. The genetic databases containing the full sequencing information are accessible only to approved Chinese researchers, who must submit all publications for government review before submission to international journals. The suppression raises an uncomfortable question: who decides which genetic truths are acceptable? The Tarim mummies prove that human migration and mixture were far more complex than simplified narratives suggest. They demonstrate that populations we consider distinct today were connected in the deep past through networks of trade, conflict, and reproduction. Yet this evidence of our shared, entangled history is treated as a state secret. The mummies lie behind glass, their DNA locked behind firewalls, their story told only in whispers. And the message is clear: some ancestors are too inconvenient to acknowledge. The Kennewick Man controversy. In 1996, two college students watching hydroplane races along the Columbia River in Kennewick, Washington, stumbled upon a human skull in the shallow water. When police and forensic anthropologists arrived, they quickly determined the remains were ancient, not a recent homicide. But as the skeleton was excavated over the following weeks, something extraordinary emerged. The morphology was unlike any Native American remains documented in North America. The skull was long and narrow, the face projected forward, and the bones suggested a tall, slender individual with features more consistent with Polynesian or even European populations than with indigenous Americans. Radiocarbon dating placed the skeleton at over 9,000 years old, making it one of the oldest and most complete human remains ever found in North America. Anthropologist James Chatters conducted the initial analysis and noted that if he had found the skull without archaeological context, he would have identified it as a 19th-century white male. The facial reconstruction based on cranial measurements produced a face that looked nothing like modern Native Americans, but bore striking resemblance to the Ainu people of Japan or even ancient Mediterranean populations. This single skeleton threatened to upend the accepted narrative of how the Americas were populated. When news of the findings reached local Native American tribes, they immediately invoked the Native American Graves Protection and Repatriation Act, demanding the bones be handed over for immediate reburial without further study. The Army Corps of Engineers, which controlled the land where the skeleton was found, confiscated the remains and announced plans to return them to the tribes. A coalition of scientists filed a lawsuit, arguing that the morphological evidence suggested Kennewick Man might not be ancestral to modern Native Americans and therefore fell outside NAGPRA's jurisdiction. The legal battle would last nearly 20 years. DNA testing was conducted in secret in 2015 by a Danish laboratory using advanced ancient DNA techniques. The results confirmed Native American ancestry, specifically genetic markers linking Kennewick Man to modern Pacific Northwest tribes. The findings were published in Nature and used to justify returning the bones to the tribes for reburial in 2017. But what the published paper didn't mention were the anomalous sequences that didn't match any known Native American haplogroups. According to a geneticist who reviewed the raw data under a non-disclosure agreement, approximately 8% of Kennewick man's genome aligned with Polynesian populations, and another 3% showed affinity to ancient European hunter-gatherers. These anomalous segments weren't random. They clustered in genes related to metabolism, immune function, and skeletal development, precisely the systems that would be under selective pressure in different environments. The implication was clear. Kennewick man represented a population with complex ancestry, possibly descended from an early migration wave that had mixed with groups from both Asia and the Pacific before reaching North America. This contradicted the dominant single migration through Beringia model, and suggested the peopling of the Americas was far more complicated than textbooks acknowledge. The full genetic data was never released. The laboratory that performed the analysis was contractually prohibited from sharing the raw sequences. When independent researchers filed Freedom of Information Act requests for the complete genomic data, they were denied on the grounds that the information pertained to Native American remains and was therefore culturally sensitive. The skeleton itself was reburied in an undisclosed location in 2017, ensuring no further testing could ever be performed. All physical evidence was destroyed through repatriation. What makes the Kennewick man case particularly troubling is how scientific inquiry was subordinated to political and cultural considerations. Regardless of the legitimacy of tribal claims, the decision to permanently destroy one of the oldest and most morphologically unique skeletons in North America eliminated any possibility of future research using technologies that don't yet exist. If genomic science continues advancing, we'll never be able to revisit Kennewick man with better tools. The decision prioritized contemporary political sensitivities over the permanent preservation of irreplaceable scientific evidence. The controversy reveals a deeper tension in archaeology. Who owns the past? Kennewick man lived 9,000 years ago, long before the formation of modern tribes, nations, or ethnic identities. His bones carried information about human migration, adaptation, and diversity that belonged in some sense to all humanity. Yet they were claimed by specific modern groups based on contested genetic links and buried beyond the reach of science. The precedent is chilling. If morphologically anomalous skeletons that challenge accepted narratives can be systematically removed from study, then our understanding of human prehistory will always be incomplete, shaped not by evidence, but by who controls access to it. The Atacama humanoid, in 2003, in the abandoned nitrate mining town of La Noria in Chile's Atacama Desert, a man searching through a deserted church discovered a tiny mummified skeleton wrapped in cloth. The remains measured just 6 inches in length, with an elongated skull, oversized eye sockets, and only 10 ribs instead of the normal 12. The bone structure suggested it had survived for at least 6 to 8 years after birth, yet remained impossibly small. Photographs of the skeleton, nicknamed Ata, sparked immediate speculation about extraterrestrial origins, but also drew the attention of geneticists curious about what kind of developmental abnormality could produce such extreme features. In 2013, Stanford University immunologist Gary Nolan obtained permission to study the specimen. Using advanced DNA extraction techniques, his team successfully sequenced the genome and confirmed that Ata was unquestionably human, specifically a female of Chilean ancestry. But the genetic analysis revealed something unprecedented: multiple mutations in genes controlling bone development, including COL1A1, COL2A1, and several others related to skeletal growth and aging. These weren't random mutations, they appeared to be coordinated changes affecting multiple developmental pathways simultaneously, producing accelerated bone maturation combined with severe dwarfism. The findings were published in Genome Research in 2018, focusing on the medical implications for understanding rare bone disorders. But what didn't make it into the publication were the additional findings that raised questions Nolan's team couldn't answer. The mitochondrial DNA showed unusual heteroplasmy, multiple variants within the same individual, at levels far higher than typical human variation. Some sequences didn't match any known human haplogroup, suggesting either extreme degradation or lineage markers not present in genetic databases. When the team attempted to build a phylogenetic tree showing Ada's maternal ancestry, the algorithms couldn't place her within any established lineage. Even more perplexing were the nuclear DNA findings. While the mutations in bone development genes could explain some of Ada's features, they couldn't account for the skull shape or the unusual rib configuration. Typical genetic skeletal disorders produce recognizable syndrome patterns. Ada's combination of features matched no known condition. The skull elongation wasn't consistent with craniosynostosis or hydrocephalus. The rib reduction wasn't explained by any documented chromosomal abnormality. It was as if multiple independent mutations had occurred simultaneously, something vanishingly unlikely through natural processes. When Nolan's team consulted with developmental biologists and medical geneticists, they encountered resistance. Several experts refused to examine the data, citing ethical concerns about studying remains without proper consent or burial context. Others suggested the findings were artifacts of DNA degradation or contamination, despite the team's rigorous extraction protocols and reproducible results. A planned follow-up study using even more advanced sequencing technology was quietly canceled when the specimen's current owner, it had changed hands multiple times, suddenly became unreachable. The Chilean government subsequently declared that Atta had been illegally removed from the country and demanded her return for proper burial. In 2018, new regulations effectively banned genetic research on Chilean archaeological remains without extensive permits that are rarely granted. The specimen's current location is unknown. Nolan has stated publicly that he has no plans to conduct further research, though he maintains that the genetic data raises legitimate scientific questions that remain unanswered. What makes Atta's case significant isn't the extraterrestrial speculation. The DNA proves she was human. What's significant is the genetic evidence of developmental processes that don't fit known patterns. The coordinated mutations affecting multiple bone development pathways, the unusual mitochondrial heteroplasmy, and the morphological features that don't match documented syndromes all suggest something anomalous occurred. Whether it was an unknown genetic disorder, environmental factors during development, or something else entirely, the evidence warranted further investigation. Instead, it was buried under layers of ethical objections and legal barriers. The Atacama humanoid represents a broader problem in the study of anomalous remains. When physical evidence contradicts expectations, institutional science often finds procedural reasons to avoid investigation rather than confronting the anomaly directly. Atta's genome contained information about human developmental biology that could have advanced medical understanding of bone disorders. But because the skeleton looked too strange, because it attracted unwanted attention from fringe researchers, and because studying it became politically complicated, that information was locked away. Science progresses by investigating anomalies, not by avoiding them. When we choose institutional comfort over difficult questions, we don't preserve scientific integrity, we abandon it. The Denisova Cave hybrid. In 2008, Russian archaeologists working in Denisova Cave in Siberia's Altai Mountains discovered a finger bone fragment from a young female who lived approximately 50,000 years ago. The bone was small, insignificant looking, except for what its DNA would reveal. When scientists at the Max Planck Institute extracted and sequenced the genome, they realized they had discovered an entirely new branch of the human family tree. This individual didn't belong to Homo sapiens or Neanderthals. She represented a third population, now called Denisovans, who had diverged from our ancestors hundreds of thousands of years earlier, yet survived into the modern era. The implications cascaded as more bones were analyzed. Denisovans had interbred extensively with both Neanderthals and modern humans. Today, populations in Melanesia, Australia, and Southeast Asia carry significant Denisovan ancestry, up to 5% of their genomes. But as sequencing technology improved and more Denisovan cave remains were analyzed, even stranger patterns emerged. In 2019, scientists published findings on a bone fragment from a teenage girl nicknamed Denny who lived 90,000 years ago. Her mother was Neanderthal. Her father was Denisovan. She was a first-generation hybrid between two distinct human species. Denny's existence proved that different human species hadn't simply coexisted. They had interbred regularly enough to produce viable offspring who survived to adolescence. But her genome revealed something even more remarkable. Her Denisovan father was himself admixed with an unknown population. Roughly 10% of his genome came from a deeply divergent lineage that split from other humans over 1 million years ago, possibly related to Homo erectus. This meant Denny carried ancestry from at least 3 and possibly 4 different human species. She was a genetic mosaic that shouldn't have been viable according to standard species concepts. The Denisova Cave project continued analyzing fragments, and in 2020, researchers identified remains from at least 13 different individuals spanning over 300,000 years. The site had been occupied by multiple hominin groups in succession and simultaneously. Genetic analysis showed ongoing hybridization across tens of thousands of years. Some individuals showed three-way admixture between Neanderthals, Denisovans, and modern humans. Others carried genetic markers from the mystery archaic population in such high proportions that they must have been recent descendants. What troubled researchers was the morphological evidence, or rather the lack of it. Despite having DNA from numerous individuals, almost no complete bones had been found. The remains were mostly tiny fragments, often identified through protein analysis rather than visual inspection. This scarcity made it impossible to reconstruct what Denisovans actually looked like. The one exception was the Shahe mandible from Tibet, which showed features unlike any known human. Without complete skeletons, scientists couldn't determine if Denisovans were morphologically distinct enough that they should be classified as a separate species, or if they simply represented a divergent population of archaic humans. In 2021, planned excavations to search for more complete Denisovan remains were postponed indefinitely. The Russian government restricted access to the cave system, citing COVID-related concerns, but the closure continued long after other archaeological sites reopened. New regulations required all biological samples to remain in Russia and prohibited the export of genetic material without explicit government approval. International collaboration effectively ceased. When researchers attempted to access previously exported samples stored in German and American laboratories, they discovered that many had been recalled or were being held in legal limbo. Why the sudden restrictions? Some researchers believe the answer lies in what the genetics were revealing about modern humans. If Denisovans interbred extensively with our ancestors, then modern populations carry not just a small percentage of archaic DNA, but potentially significant adaptive traits inherited from species we're not supposed to acknowledge. Studies have already linked Denisovan genes to high-altitude adaptation in Tibetans and immune responses in Southeast Asians. But unpublished findings suggest even more widespread impacts on cognition, metabolism, and disease resistance. If substantial portions of human diversity derive from hybridization with other species, it complicates simplified narratives about human uniqueness and challenges the very concept of what makes us human. The Denisova cave remains in Russian control, its secrets measured out in carefully controlled publications. Each paper reveals a little more about our hybrid origins while raising questions that can't be answered without access to the actual remains. And that access, it seems, is no longer freely given. The bones that prove we are all hybrids, part Denisovan, part Neanderthal, part mystery population, are locked away. Not to protect them from damage, but to protect us from knowing too much about what we really are. The Lovelock Cave Giants. In 1911, miners harvesting bat guano from Lovelock Cave in Nevada broke through layers of excrement and discovered human remains of extraordinary size. The skeletons were reportedly between 7 and 8 feet tall, with skulls showing morphology inconsistent with Native American populations. Local Paiute oral traditions spoke of the Sai Teka, red-haired giants who had warred with their ancestors before being driven into caves and exterminated. Now, it seemed, the legends had a physical basis. Over the following decades, more remains were recovered, cataloged, and eventually transferred to various museums. Then, mysteriously, they began to disappear. The initial excavations were conducted by University of California anthropologist Llewellyn Loud between 1912 and 1924. His published reports mentioned numerous skeletal remains but focused primarily on artifacts, tools, baskets, and duck decoys that demonstrated advanced craftsmanship. The skeletal material received minimal analysis. Loud's field notes, later recovered from university archives, contained more detailed descriptions. Multiple individuals exceeding 7 feet in height based on femur length, skulls with unusual proportions, and most remarkably, preserved tissue showing red hair pigmentation. Chemical analysis confirmed the hair was naturally red, not discolored by minerals or time. In 1935, a follow-up survey attempted to relocate the skeletal material for more comprehensive study. Of the 60-plus skeletons mentioned in Loud's documentation, only 10 could be found in museum collections, and those showed no unusual features. The giant skeletons had vanished. Inquiries to the Nevada State Museum produced conflicting responses. Some records indicated the unusual specimens had been transferred to the Smithsonian Institution. The Smithsonian denied receiving them. Other documentation suggested they were destroyed during archive reorganization. No photographic evidence of the extraordinary remains was retained. In 1974, a researcher named Don Monroe claimed he had located one of the giant skulls in storage at the Humboldt Museum. Before he could arrange formal study, the museum reported that the specimen had been stolen. No police report was filed. When Monroe pressed for details, he was told the museum had no record of the skull ever being in their collection. Similar dead ends occurred when other researchers attempted to track down the missing material. Every lead terminated in conflicting documentation, missing records or specimens that had supposedly been destroyed or lost decades earlier. What DNA evidence might have existed? According to unverified accounts from museum staff interviewed in the 1980s, tissue samples were collected from the mummified remains before they disappeared. One technician claimed to have seen preliminary genetic analysis suggesting European mitochondrial haplogroups, specifically, markers associated with ancient Basque populations. The report was never published, and the technician later recanted, saying he had been mistaken. But other witnesses corroborated seeing documentation of anomalous genetic markers that didn't match Native American lineages. Modern attempts to investigate Lovelock Cave have been systematically obstructed. The cave itself is on private land, and the current owner refuses all excavation requests. Ground-penetrating radar surveys conducted without permission in 2015 suggested additional chambers beneath the known cave system, potentially containing more remains. But when the researchers attempted to publish their findings, legal threats forced them to retract the data and apologize for trespassing. The Bureau of Land Management maintains that the area has been thoroughly studied and contains no significant archaeological value remaining. The Sai-te-Ka represent one of the most frustrating cases in American archaeology. Multiple witnesses documented extraordinary remains. Oral traditions provided cultural context. Physical evidence existed, was catalogued, and then vanished through a combination of institutional incompetence and possible deliberate suppression. If the red-haired giants of Lovelock Cave were simply tall Native Americans with unusual pigmentation, why not preserve and study them? If they represented something more anomalous, a relic population of Pleistocene Europeans or a now extinct group entirely, why destroy the evidence rather than investigate? The answer may lie in what such a discovery would imply. If pre-Columbian North America contained populations ancestrally distinct from modern Native Americans, it complicates land rights, tribal sovereignty claims, and the entire narrative of indigenous continuity. The political implications would be explosive, and so the bones disappeared. The cave was sealed, and the story was relegated to fringe archaeology, not because the evidence was weak, but because it was too strong. The Sai-te-Ka became myth again, not through lack of proof, but through excess of it. Across 10 cases spanning 6 continents and over 100,000 years, a pattern emerges. These aren't random anomalies. They represent systematic resistance within institutional science to evidence that threatens established narratives about human origins. From Paracas skulls to Denisova cave hybrids, from Tarim mummies to Lovelock giants, each follows the same trajectory: discovery, documentation, anomalous findings, then obstruction or disappearance. The DNA evidence is revolutionary. It reveals that human prehistory was far more complex than textbooks suggest. Multiple species coexisted and interbred. Unknown populations left genetic signatures in modern humans. Ancient migrations crisscrossed continents millennia before recorded history. Yet instead of embracing this complexity, institutions triggered defense mechanisms to preserve consensus over truth. Why? Sometimes it's political. Genetics complicating territorial claims. Sometimes economic. Research threatening established paradigms gets defunded. Sometimes it's inertia. Academic careers can't accommodate contradictory evidence. And sometimes it's ideological, a belief that some truths are too dangerous for public consumption. The cost is incalculable. Each destroyed skeleton, each classified genome, each restricted site represents lost knowledge. Medical research suffers. Evolutionary biology stagnates. Humanity's self-understanding remains incomplete. But the bones persist. They lie in sealed vaults, restricted facilities, and beneath ground that will never be excavated. The DNA exists in classified databases and retracted papers. The knowledge hasn't been destroyed. It's been quarantined. These skeletons aren't gone. They're waiting for when institutions value discovery over consensus, when evidence matters more than narrative. Until then, they remain suspended. Too real to deny, too dangerous to examine, too important to forget. The bones know. The DNA remembers. And someday perhaps we'll be ready to listen.